So now that we know how to measure the rates of enzyme-catalyzed reactions, we can now start isolating variables and see how they affect the rate of enzyme-catalyzed reactions. This video is going to be focused on the effect of temperature on enzyme action. So the temperature has actually two effects on enzyme-catalyzed reactions. The first thing is that when you increase the temperature, you are adding energy in the form of heat. This energy is going to transfer to the molecules as kinetic energy. Kinetic energy is the energy of movement. So the more you increase the temperature, the more you are adding heat, the more energy is being transferred to molecules as kinetic energy, so the faster these molecules are going to collide, which means now the substrate and the enzyme are going to meet with each other more often and faster. So this is actually going to increase the rate of the reaction. However, the increase in temperature also causes some bonds, like hydrogen bonds and others, to break inside of the enzyme, whether in the active site or in other sites. And this causes the enzyme to change its shape. So at some point, if the temperature starts to increase too much, the bonds are going to break so much that the shape of the enzyme is no longer recognized by the substrate. The substrate can no longer fit in the active site of the enzyme. There is one temperature, if you reach it, in humans, for example, this is 60 degrees Celsius, you completely destroy the enzyme. The enzyme is now called denatured. You denature the enzyme at this high temperature. And once you reach the denaturation temperature, once the enzyme is denatured, you can no longer go back. Even if you decrease the temperature, the enzyme is lost forever. So the effect of temperature on the enzymes is a balance of both of these factors. First, it's going to increase the rate of the reaction, but also you have to stop at a certain point, because if you exceed this point, you're changing the enzyme's shape so it can no longer act on the substrate. And if you reach even higher points, you might denature the enzyme. So for example, this is a graph measuring the rate of the reaction in the human body as a function of temperature. So this is a human enzyme. We know that the human body is always at a temperature of 37 degrees Celsius. This is not actually the optimal temperature for enzymes in the human body. The optimal temperature for enzymes in the human body is 40 degrees Celsius, as you can see here in the graph. As the temperature increases from 0 to 40, the rate of the reaction catalyzed by this enzyme increases to reach a maximum at 40 degrees Celsius, whereby it starts decreasing when we increase the temperatures further than 40 degrees Celsius. So 40 is the optimal temperature, and any temperature beyond that is going to start decreasing the efficiency of the enzyme because of changing its shape. The substrate is no longer fitting. And at 60 degrees Celsius, we see that the reaction stops completely. This tells us that here, the enzyme is denatured. So here it's no longer decreasing the rate of the reaction, it's stopping the reaction completely, because the enzyme is completely denatured here. So if 40 degrees is the optimal temperature for enzymes in our bodies, why do we maintain our bodies around 37 degrees Celsius? Well, around 37 degrees Celsius, even though it's not the optimal temperature for these enzymes, it is not a bad temperature. It's a temperature at which these enzymes can still function efficiently. So the cost of keeping your body at 40 degrees Celsius is so high, because this is energy that you will constantly have to supply your body with, through food, for example, to keep burning this food and making this heat. So maintaining a 40 degrees Celsius is very energy demanding. Maintaining 37 degrees Celsius is more feasible. And also, this gives you room when you become sick. Your temperature can rise to 40 degrees Celsius without denaturing your proteins. Remember that enzymes are not the only proteins in your body. There are other proteins that can also be denatured at high temperatures. Some proteins in your body are denatured beyond 37 degrees Celsius. So this is basically a compromise between enzymes and other proteins of the body. So even though 40 degrees is the optimal temperature for the enzymes in the body, it is too costly and too risky to maintain your body at 40 degrees, and that is why our bodies are maintained at 37 degrees Celsius.